visiting seminar speaker here today in the EEB department. So Jenna's research focuses on plant ecophysiology, particularly focusing on extremophiles in the desert. So Jenna started working on extremophiles during her master's thesis at California State University of Los Angeles, where she did a bunch of work out in the Mojave Desert, focusing on moss extremophiles. She then went on to get her PhD in integrative biology at the University of California at Berkeley, where she sort of continued her work focusing on Cetrichia mosses and getting into some lab-based experiments in ecophysiology, some in the field, and then also some transcriptomics. She's currently a postdoctoral research fellow at the Smithsonian in the Data Science Lab. So if you're interested in sort of Smithsonian-oriented things, you should definitely chat with Jenna, because the Data Science Lab, as I understand, is sort of located under IT, which is kind of an interesting sort of funding avenue to get involved in as a biologist. And so she has super diverse lab mates who are in all sorts of realms. So with that, it's my pleasure to introduce Jenna. I met her through the bryology groups, of course, when I was out at UC Davis in California. And I'm excited to hear about the effects of full sun spectrum sunlight on desert mosses. So take it away, Jenna.
Thank you, Jessica. Thank you, and thank you all for having me. If I'm too quiet, too loud, please just let me know. It's hard to tell from up here and through Zoom. But yeah, so I'm excited to talk to you about most of this work is part of my dissertation or side projects I did in grad school. I'm about a year into this postdoc, and I'll post some of like the current things, but mostly it's I don't really have any new results to share yet. So this is the stuff that's that I'm sort of continuing with. But before we get into like the specifics of what I'm working on or have been working on, I have to introduce mosses. This is a diverse group. I know the department is very diverse. And I would like to point out that mosses, while plants, they're not like other plants. They do have, like all land plants, they have this alternation of generations life cycle that all of us have learned at one point or another. But so same life cycle, but it looks a little different. For one, mosses are haploid dominant. So their main body, their gametophyte body is haploid. And what that means for sex determination in their life cycle for the species that have separate sexes is that that sex is determined by a single chromosome. So it's a UV system where U is the female chromosome and V is the male. And you can see that when after sexual reproduction and the sperm and egg fuse, there's a sporophyte which has both U and V chromosomes and is unsexed. Sexual reproduction, I don't know if you can see that. Sexual reproduction happens by flagellated sperm. Okay, that's different from many plants we're probably familiar with, and spores. And I study plants that occur in dry lands and where there's not a lot of water availability, so that becomes really important. Therefore, all mosses can reproduce asexually, and it's very common in all mosses, and particularly in desert ones. And cloning can occur by fragmentation. You can chop up a moss and it grows new mosses. Specialized asexual propagules called gemmy, or by selfing for those that kind of have they're hermaphroditic, you could say. Other ways that mosses are not like other plants are the water relations. They're very, very different in important ways. Mosses are poikilohydric. This is a very weird word that means that the cellular water content equilibrates to the external water content. And this is opposed to homeohydry, which plants try to maintain their preferred water status at all times. This is for the animal people in the room. I talked to some people who study ectotherms, somewhat equivalent, analogous to kind of ectothermy and endothermy, but with water, but some differences, of course. And sort of a corollary then of being poikilohydric is that when it's dry out, the mosses are dry. And this means that mosses are desiccation tolerant. Desiccation tolerance is a spectrum, and so some are more desiccation tolerant than others, and they can be hardened to it. But fundamentally, mosses can dry out and survive it, and that's drying or desiccation tolerant. Desiccation tolerance, though, is pretty specific definition that they can lose all cellular free water and recover normal function after rehydrating. And the mosses that I work with are like experts in this. But this drying, re-wetting process is, most mosses you'll see it, and you may be familiar in kind of the forests around here. The Smokies, if you go out after a rain, everything looks green and livened up, and that's because of this same mechanism. But in desert plants, it's extreme. When they're dry, this is the same species, Cetrichia ruralis, and on the right is it's dry, and it might look dead to you, but really it's just dried out and kind of dead maybe because it's not really living. But you put water on it, and it looks like that on the left, and looks like a normal moss. Cetrichia is a genus of moss that has about 90 or 100 named species, many of which are dryland specialists, so including like arid grasslands and steppe. And many of the members of Cetrichia are important in biological soil crusts, which are the surface community in drylands, the soil surface community, excuse me. Cetrichia has a wide range of morphological and physiological variation, though you can find Cetrichia in all kinds of habitats, even though there are a lot of these dryland specialists. And just as a plug, there's a whole NSF Dimension of Biodiversity project we're sort of finishing up right now that kind of explores all the variation within this group. 3dmoss.berkeley.edu if you want to check out more. But as I kind of mentioned, some of the really most desiccation tolerant plants are in this group. And so the rest of my talk is pretty focused on Cetrichia. The rest of my research is. And yeah, most desiccation tolerant plants known, and that includes like the resurrection ferns and other things that are pretty impressive as well. So I explained like what desiccation tolerance is all about, but I feel like it's important to see what this looks like. This is Cetrichia Canada. Don't know how to get it to play. I'll try it like this. Oh no. Okay. Oh, this might do it. Yeah, okay. All right. So Cetrichia Canada. This is what it looks like when you find it like dry in the desert. There we go. And I sprayed some water on it, and you can see the leaves are opening up, and it's turning. It's not an illusion. It's turning green before our eyes. And this is in real time. Maybe I should try that again. It's in real time. It really goes that fast. Technology. You hear it for a second. Okay, let's just let us watch it. Move on. All right. It really is that fast. So part of the desiccation tolerance mechanism is that it can recover really quickly. These real pros at it can recover really quickly, and that's important for when in the desert, when the rainfalls are brief and the water may dry up pretty soon afterwards. They can get back to that positive carbon balance pretty quickly. At least that's the goal. Desiccation tolerance, though, as I mentioned, is a whole suite of, it's a spectrum with a whole suite of traits, and some of those have genomic signatures. So I just want to point out in Cetrichia Canada, there are some mysteries that are associated with desiccation tolerance, or we have evidence to think so. For one, there are 1,500 orphan genes in this species. Maybe they're not all related to desiccation tolerance, but we don't know what they're doing or what they're related to. And four gene clusters within there are associated with desiccation from our experiment. So there are a lot of real novel genes that are involved, and some of the examples of those are genes. There are also genes that are known in other processes that are important in desiccation tolerance. ELIPS, early light inducible protein. This was known to be involved in light when it was first characterized, very important in desiccation tolerance, and late embryogenesis abundant LEAs that are also really important in desiccation tolerance, and as far as we know, not involved in embryogenesis here. So interesting, these genes are important for desiccation tolerance across land plants, actually. So it continues to be true in Cetrichia. And in Cetrichia Canada, there's a huge expansion of ELIPs, so they're playing a major role here. Some implications of desert moss, of life as a desert moss, though, that, as I mentioned, there are things that really, really stand out. There are physiological implications, such as frequent and prolonged desiccation. You can imagine in places like the Mojave, in the Western Mojave in particular, where I did my research, summer rains are infrequent. And so you can have several months on end with no water availability. And even in the winter, there are drought years in which rainfall is infrequent. So you can have repeated desiccation events throughout the winter as well. Throughout the summer, plants must tolerate excess light, even while dry. And that is important because they can't use any of it. So when they're hydrated, there's probably much more light than they can use. Also, it's a real intense sign, but they can't use any of it when they're dry. There are implications for growth and population dynamics. You can imagine with such brief watering events, they don't have very many opportunities to grow. And this is true, even when they do grow, it's relatively slow. For example, some populations of Cetrichia Canada in the Mojave Desert that were measured grew on average 0.36 millimeters a year, so pretty small amount. And in populations, sex ratio biases are extremely common in desert mosses. They're common in a lot of bryophytes, but in desert mosses, they're pretty extreme. There can be as many as, in all the populations studied so far, as many 5 to 20 females for every male. And some populations are entirely female, we're finding. And so it could, the max extreme. In all the desert mosses, it's a female bias. And that tends to be most mosses that have a sex bias, it's female biased for reasons we don't yet understand. I wanted to throw this quick plug in here for a new tool that just published to help so sexual reproduction is rare in these plants. And so many Cetrichia Canada stems, you cannot tell their sex by looking at them because they don't make their sex organs. So here's a little schematic of 3, what they would look like. The top has archegonia, the female sex organ. This middle has antheridia, male sex organs. But this one here has neither. And so we don't know if it has that U or that V chromosome. Previously, you would have had to do some intensive sequencing to find that out. And that's what I did for my master's. But out of that project, we developed a easy to use PCR tool, restriction fragment length polymorphism, to tell those apart. And they work in other members of Cetrichia too. So it's pretty fun and easy. And I think it would make a great student project if there's any interest in that. To survey new populations, because we've only done a few, and so far there's a lot of variation and we don't know why, and kind of the more data the better. So this is just some details on how it works, but I would be happy to talk to you about any of that if you're interested. Oh, and this paper has a case study using legacy collections. So we use it on mosses that were collected more than 20 years ago, and it was effective. So it can be it can work well on older specimens too. Okay. So it was doing some of this research on population genetics that Jessica mentioned and that I cited there that I actually came across a unexpected finding, and this was in the Sheep Creek Wash in the San Gabriel Mountains, the high elevation Mojave Desert of California. My collaborator at the time, my master's thesis advisor, Kirsten Fisher, picked up a quartz rock and we saw moss underneath it. And that was strange because mosses, being plants, need light. And this rock doesn't look particularly translucent. It didn't seem like a plant would be able to live under that, but it was. But we didn't know, is this like a one-off kind of freak occurrence, or is it more? So we went to the literature, as you do, and there was only a single report of mosses growing under rocks in the Mojave Desert from 1989, and it was just a report of a single patch at a different part of the Mojave and a different type of rock. So, and with no subsequent analyses, and we were left with a lot of questions about what we just found. Is it a thing, or is it not? I continued searching the literature and found a whole wealth of research on what are known as hypoliths. Cyanobacteria hypoliths are relatively well studied, and including in the Mojave Desert. And some of those hypolithic cyanobacteria studies referenced the presence of mosses under rocks in Antarctica. So I knew that, okay, this is a thing. It's in Antarctica. It's here a couple of times at least. It's like a real phenomenon. So we designed a study. And oh, I forgot to mention, there's actually one obligate hypolith that we know about in endemic to Kansas. Yeah, I haven't seen it yet, but I would love to. So that's pretty neat. It also grows in a semi-arid arid landscape, and I would love to find out more about the climatic conditions in those habitats. So we designed a study to learn more about this Mojave Desert hypolithic moss habitat because there was so little known. First, I wanted to understand the community composition. When we were just out there poking around, I could see that there was more than one species that occurred under the rocks. I wanted to know about the quartz rock light transmittance, how much light was getting through these rocks. And I wanted to know about the microclimate under there, and in particular, temperature and relative humidity, both of which are pretty important for Ekwealor moss. So we selected the rocks, measured the thickness, and deployed eye buttons under the rocks and on the surface to compare the soil surface microhabitat. Community composition, to identify species in moss, you have to do some microscopy. They have pretty small characters that are very precise. So this is kind of what those data look like. This here is a picture of Cetrichia Canada. You may have seen those white hairs that we're showing on the ruralis too. That's a leaf tip of Tortula inermis. That both of those ended up being pretty common. And these are what these plants look like kind of as whole shoots. So these turned out to be pretty big players in the hypoliths. There was also the species Bryum argentium. This is a cosmopolitan species that kind of occurs everywhere, including in the deserts, but it's pretty low abundance in the desert. So it's kind of like everywhere in small amounts here in the desert anyway. And just to visualize the species composition results, the purple up here is the surface, and the green is hypolithic, and then the three species. So you can see just a big chunk is still Cetrichia Canada, the dominant species of this site. But Tortula inermis was much more abundant under the quartz than it was on the soil surface. And as I mentioned, Bryophyter is pretty uncommon and didn't occur at all as a hypolith. So just looking at the species distribution told us, okay, the composition is distinct. The proportions of species and where they occur appears distinct in the two habitats. But it was puzzling because being a desert, I went into it maybe with the bias that the quartz rock probably makes it cooler under the rock, and yet this Tortula inermis that seemed to be preferentially growing under the quartz rocks is a moss that's found down in lower elevations, and in the desert, lower means hotter. It's a plant that can tolerate hotter temperatures and yet is kind of hiding out under these rocks at this temperature. So it was a puzzle to me that eventually I think I figured it out, but I'll tell you how. But this was the question: why is it nearly restricted to quartz hypolithic microhabitats at this elevation? It doesn't make sense. So the temperature results maybe help. This graph is a line of the temperatures, and the light blue on the top is the daytime, and dark blue is night. The data loggers were deployed from September to March or late February. And when it goes above the zero, it's the difference in temperature there between surface and hypolith. So when it goes above the zero, it's cooler under the rock, and below, it's warmer under the rock. And so right away, you can see this strong day-night effect, where during the day, the rock is a cooler place underneath the hypolith, and at night, it's warmer. And that's sort of consistent on what a plant or an organism might want in a desert, right? Deserts are hot during the day and cold at night, and this appears that it's buffering that. It makes it a little bit cooler during the day, a little bit warmer at night, and buffering the extremes. You can see there's two areas around December where the difference between surface and hypolith there's no difference. Sorry, between day and night is no difference, but it's a little bit consistently warmer under the rock. It's just below zero. I don't know if you can tell. And those are two snowfall events. So when they were covered in snow, day and night were the same, but it was consistently warmer under the rock. And also this kind of buffering effect was overall stronger in the warmer months, less pronounced in the winter months, but still there. So, and the differences we're looking at here, mean differences is 2 degrees Celsius in each direction. So what this told me is that actually the rock provides warmth. And so if this Tortula inermis is more adapted to warmer temperatures, it's less tolerant of cold. Kind of makes sense that you find it under the rock at this elevation where it's getting protection from the cold temperatures. So that was a bit surprising to me because, you know, I think of deserts as hot places, but they're also cold places. They can be. The other thing I noticed while looking at these is that the Cetrichia Canada, the dominant moss, looked different. So the one on the right is a typical moss shoot from this habitat, from the desert, from the soil. And the one on the left is a very typical hypolithic Cetrichia Canada. It's longer, it's greener, and it just doesn't look right. Just take my word for it. So first I thought, okay, the length is a huge difference. So I measured the lengths, and indeed they grow longer shoots under the rock. 62% longer, in fact. Now, why could this be? Well, maybe desiccation tolerance. Plants can only grow when they're wet, right? They can't grow if it's dry. And so hypoliths may have longer hydro periods. It's difficult to get that data directly, when they're hydrated, but I think looking at relative humidity can tell us something about how long water can persist under the hypolithic habitat. So that's what I did. Similar to the temperature graph, with the day-night. And in this case, above the zero is more humid under the rock, and below is less humid under the rock. So in the winter months, there's a strong a day-night effect, right? During the day, it's more humid under the rock, and at night, it's less so. In the winter, however, it's always more humid under the rock, day and night. Let's see. So what this shows is that during the winter, there's maybe no buffering effect happening, but this is the growing season and it maybe doesn't matter. But during the warmer months, there's a buffering from desiccation. It appears that during the day, when the sun is out, when they can do photosynthesis, there's more relative humidity, which may mean longer hydro periods, more time to grow, and therefore explaining why these Cetrichia Canada shoots are bigger under the rocks. But it still was sort of a mystery on how they're doing it, because like these rocks are like really white. You know, they're not, there are quartz crystals that are super translucent, but these are not. They're milky quartz. So there's actually geological studies on this sort of thing and how much light transmits through these rocks, and it's as a function of quartz thickness. Actually was on these exact kind of quartz from the Mojave here, and according to the model, our average quartz was 2.6 millimeters, I think, or sorry, centimeters. And for that thickness, it would be 1.2% of light reaching the moss. Now that was an average, and I was just using this equation. So I measured, I couldn't take all those rocks, it'd be a bit destructive sampling, but I took two kind of to represent the range and measured empirically just how much light gets through, and found similar range here, 0.4% on the thicker rock and 4% on the thinner. So very small amounts of light getting through. And this is maybe not that surprising, because lab studies on mosses, even those that live in the desert, find that they're fundamentally adapted to low light levels. They just can tolerate excess light. But in fact, during the growing season, it's usually overcast. It's not that sunny when they're awake. And when it's too sunny, they tend to be checked out and dormant and desiccated. So it lines up. They don't need that much light. They don't want that much light. Another thing from this geology study, though, was this is the equation of the line I just here, and the solid line is how much is absorbed. The dotted line is how much is transmitted as a function of wavelength. Here it's plotted as wavelength, and one thing that it's a bit hard to tell, but this gray transmitted line, smaller wavelengths transmit less than larger wavelengths. And this was interesting to me because smaller wavelengths is where ultraviolet radiation lies. And so they're actually getting not just less light, but a different light quality, different proportions of different wavelengths. And you know, I had no idea if that was good or bad, but it was apparent. But it got me thinking that this color thing I was saying seems kind of like a suntan effect, right? They have lower light, maybe, and less UV. And so I wanted to follow up and why they look different, and is it a suntan? And yeah, so what is inducing this color here? And in fact, there are mosses that get suntans, that's known. Many mosses are known to make sunscreens. Most of this research is out of Australia, and looking at mosses in Antarctica with the ozone hole. And UV can induce all kinds of flavonoid compounds. In fact, if you take these Cetrichia Canada plants, even these dark brown field ones, and grow them in the lab, they stay bright green like this, and they never get this dark brown coloration. So I had a lot of reasons to think that this coloration is a plastic suntan effect. But I had to test it. So is the dark color due to ultraviolet radiation? So I set up an experiment in the Mojave Desert to try and tease this apart. But before I get into that, real quick, here I wanted to point out this light quality thing. Blue and red light, as you can see here, are important for photosynthesis. Yeah, you can kind of see my mouse. The chlorophylls absorb blue and red light primarily. And ultraviolet light down here does get absorbed by some things, but it is known to be damaging to plants. It can be damaging, although it has some positive effects on plants as well. Like, for example, it can induce defenses against pathogens and herbivores and other stressors. So generally, UV exposure to UV is sort of associated with tolerating stresses in other plants. And some of these here are, you can see carotenoids and other accessory pigments. Important to note that carotenoids can play multiple roles. They can absorb light, as you saw on the last slide. They can function in antioxidant activity, and they can function in thermal dissipation of excess light. So when there's too much light that the plant, more light than the plant can use in photosynthesis, it can turn it around and send it off as heat and get rid of it. So the same molecule can do those different functions. Okay, back to the experiment. This was at the Sweeney Granite Mountains Desert Research Center in Southern California. And I really just wanted to answer these two questions that had to do with tolerating ultraviolet radiation. Does UV induce a sunscreen? And does it affect photosynthetic biology? So the experimental design was to filter ultraviolet radiation from Cetrichia Canada in the natural habitat. I wanted to do this first in the natural habitat because it's difficult to impossible to recreate some of these extreme stressors in the lab. The sun, for example, you just can't recreate that. And I wanted to use this opportunity to tease apart the difference between the effects of full intensity sunlight and UV. And then after filtering out UV from natural light, I wanted to measure the effect on color, the photosynthetic pigments, and the transcriptomes, as well as recovery from desiccation as measured by chlorophyll fluorescence, which is a proxy for looking at photosynthetic performance, you could say. And this is what the experiment looked like. I had windows, UV filtering windows, UV transmitting windows as a control, because to control for the effects of the window itself, and also a third site reference with no window at all, just to see what was the effect of the window treatment. In this photo, it's pretty hard to tell because these guys are cryptic, but all these black little dots are Cetrichia Canada in here. And I had 20 triplets like this, and I left them in place for a year. So after checking on these windows every month for a whole year, they stayed in place. It was a miracle. And the first thing I saw at looking at the plants was that UV radiation filtering causes plants to appear greener. So this on the left is UV filtered, and on the right is the totally unmanipulated site reference, and the middle is the UV transmitting control. And that jumped out at me right away. This is you can find green ones like this in the wild, but it would be like if it's in a really shady spot or something. Like, this is pretty extreme. But I didn't trust my eyes, so I did some photo analysis and quantified the pixels green and red pixels, and it was real. I think there's genuinely a higher proportion of green pixels with UV removal. But being greener, green is chlorophyll, and I wanted to know if greener meant less of another pigment or more chlorophyll. And so I quantified chlorophyll in these 3 treatments. And even though the UV filtered plants appear greener, they actually have slightly lower proportion of chlorophyll compared to other pigments, in particular chlorophyll B there. And in fact, the chlorophyll A to chlorophyll B ratio is important in understanding kind of the status of the plant. And a high chlorophyll A to B ratio is considered a high light response in plants. It's known to be a high light response. So it was kind of weird. You take UV away and the plants respond as if they are getting too much sunlight. It didn't quite add up. They respond in a high light response. Coming back to that later, I then moved on to the rest of the color difference. Is the color difference due to other pigments? So I looked at a host of other photosynthetic pigments, and several of them were not significantly altered by ultraviolet filtering. Using kind of natural samples in a field setting, there's a lot of genotypic variation possible, microhabitat variation. So it's possible that some of this is just par for the course, and the trade-off of doing a field experiment is lower sensitivity in detecting differences, which is totally fine. But some things kind of rose above that variation and still jumped out as a difference. In particular, neoxanthin and zeaxanthin are 2 accessory pigments that are, well, zeaxanthin higher and neoxanthin lower with UV filtering. Again, both of those are highlight responses. So removing UV, another clue, you remove UV from the habitat and the plants respond as if they're getting more sunlight. Wasn't adding up yet. So I went to the transcriptomes looking for clues. For the transcriptomes, I just sequenced the total RNA from the 2 window treatments. And 2 of the top 10 most differently abundant transcripts were involved in flavonoid biosynthesis. I mentioned that flavonoids are known to be UV sunscreens in mosses induced by UV radiation. And the names of these are here. This first one is involved in actually both of these here are involved in breakdown of flavonoids. So when UV filtered plants have more of this transcript, it seems that means increased breakdown of the flavonoid. So, UV filtering, it increases flavonoid breakdown, therefore reducing flavonoid content, which was my clue that the pigment was in fact a flavonoid. Finally, I looked at this recovery from desiccation in a photosynthetic context. And to do that, it required this lab experiment with the field manipulated samples. Field collected plants, which are collected dry, they're dry most of the time that you're in the field, were allowed to recover in a growth chamber. And by recover, I mean just low light, humid conditions, and access to water. They're kept fully hydrated. They recovered for 192 hours, which was like 8 days. I left them in there, and at 3 time points, measured photosynthetic quantum efficiency, this Fe over Fm, which is a metric that tells you how much of the incoming light is being used for photosynthesis. And so I measured, the first measurement was at 30 minutes post rehydration, so basically straight out of the field. And you can see the same 3 treatment groups here: UV filtered, UV transmitted, and site reference. And FE over FM is this normalized ratio, so it goes from 0 to 1. Although the sort of maximum possible efficiency is like 0.85, because it's never going to be like perfectly efficient. But low, these were extremely low straight out of the field. I mean, these readings were like, generally, if you see readings this low, the plants are dying. This will not survive. So I thought I was doomed. But thankfully, I checked again the next day, and they had recovered up to at least 0.6. And interestingly, the 3 groups had this pattern where UV filtered plants had a lower FE over FM, transmitted in the middle, and site reference the highest. If you might have noticed that colors were that way too. The site reference were totally dark and the UV reference totally green. The transmitted window was kind of in the middle, sort of like this. But the important comparison here is these first 2 treatment windows, because they're the ones that only differ by UV radiation. So that was interesting. I had thought, okay, if UV is a stressor, you take UV away from these plants and they're going to do better. I was convinced that this is what the results would be. I didn't even plan for another possibility. And this totally surprised me. I took UV away for a whole year, and the plants are, by all ways that we know how to interpret these data, are not recovering as well from desiccation. 7 days after that, the pattern persisted. UV filtered plants still, they're doing a lot better, but still having that same stepwise pattern in which they're not UV transmitted is better than, and site reference, the totally unmanipulated, are recovered the most. So this was, like I said, a surprise. Removing UV appears to be the stressor, in fact, not the UV itself. And one clue I had was, well, if I put it together with the other highlight responses, it seems that it's possible that UV radiation is a cue for highlight for the plant. So if the plant sees a whole bunch of UV, it says, hey, there's going to be a lot of light. Let's prepare for a lot of light. And without that cue, it doesn't adequately prepare for the incoming light. I separated those 2 things that in nature would always correlate. So I think that's what's going on, but I have more ideas to follow up that way. Another thing that's going on here, though, is just looking at this pattern in itself, going from almost 0 FE over FM to like fully recovered over a week. Even in these totally unmanipulated site reference plants, this is happening. And that in itself was a huge surprise. Like I mentioned, that low was would normally be a plant on its way out. And I forgot to mention that I collected these in spring, late spring. They had a good winter. It was not a drought, not an excessive drought year. I guess we're always in a drought now in California, but it wasn't excessively droughty. There was a lot of rain, a good year. And this is when these plants should be at their best, right? They had this whole growing season. They should be at their healthiest. You would expect at the end of summer, they might be kind of doing rough. But according to these data, this was like a plant that can barely do photosynthesis. It was not looking good. But it turns out that, oh, and it had only been 2 weeks since its last rain. So, oops, sorry. It was not even, they hadn't even dried for that long. It turns out, though, if you look at this FE over FM is this ratio. And if you look at the underlying components of this compound metric, that it's composed of FM and FO. It can give you some clues about what's driving the change in FE over FM. And this was a change in FE over FM driven by FM, maximum fluorescence. There's a lot of technical details here, but take my word for it at this point. I'd be happy to talk about it more because there's too much to go into right now. But a change in FE over FM driven by FM like this is indicative of this process called a release of sustained thermal quenching. I mentioned how those carotenoids can release excess light as heat. Well, some of them do that so strongly that it takes days for it to recover, for it to fully let go and start doing photosynthesis again. And so that's what these data were showing, is that these plants had a way of fully shutting down photosynthesis such that even when they were rehydrated, they weren't turning it back on right away. It would take 8 days for them to turn it back on up to what is considered efficient levels, really, 0.8 at least. And that UV was, the removal of UD was hindering that recovery. This mechanism that I'm describing is not known in these mosses, but is pretty well characterized in overwintering evergreens in high elevation habitats. So similar to mosses, they go through an entire season where there's a high light and little or no water availability when there's snow and the ground is frozen. These plants still have all their chlorophyll, they have all their leaves, they're evergreen, but they can't use any of that light if they don't have water. So they go through this sustained thermal quenching where it's a very extreme shutdown of their photosynthesis as a protective mechanism. And it's one that takes days to turn back on. And so what I think, I only have this one point I collected in spring. So I need to sample throughout the year to test this hypothesis. What I think is happening is the same thing as the overwintering evergreens, that the plants enter the summer in a completely shut down state, and in fact aren't turning things back on as quickly as they do in the winter, for example, when they are getting rain all the time. So it's I think, pretty cool finding, but it's not complete yet. What we know now is that it happened this one time, but we need to test for a seasonal variation. And it's possible that ultraviolet radiation is the seasonal cue that tells plants what season it is, these plants. Ultraviolet radiation is from what I understand, a fairly reliable cue for what time of year it is. And there are other plants that use UV as a seasonal cue, such as for flowering times and other things. So this would be consistent with those data. So yeah, so that's one of the things I hope to work on soon. A couple more transcripts that were important in these window treatments, so were related to oxidative stress. 2 more of them were polyamine oxidase and osteolipid desaturase-like. Both of these have functions, and again, this is transcriptomics. I should mention, none of these have been characterized in this species. So these are like hypothesized functions for the species. So plenty of avenues for possible research to follow up on these. But as a clue, I looked at these as clues of what might be going on. As you can see, both of these transcripts are known to be associated with stress responses. And again, you remove UV radiation and these stress transcripts increase. So this is another clue that the plants with UV filtered out are not doing as well. And UV removal in the natural habitat increases stress for this species. Okay, so getting towards the end here, I did a couple of experiments in the lab to try and sort of narrow down the number of variables, where I grew Cetrichia Canada and another species, but and exposed them to different amounts of ultraviolet radiation to see kind of a very narrow what does it do when it sees UV. So for this experiment, I grew them up in a UV-free environment in the growth chamber and exposed them for up to 30 minutes, 0, 10, and 30 minutes of broadband UV radiation to cast a wide net, UVA, UVB. and to plants that had never seen it before. And I performed transcriptomics on those again to compare to these field findings. And in these transcripts, so I should mention, I look for differentially abundant transcripts as indicators of what might be going on. I looked for some of these candidate gene families like Ellipse and Leas that are really, really well known to be involved in desiccation tolerance. Because we were seeing this inhibition of recovery with UV removal, recovery from desiccation. And so I had a hypothesis that that somehow removing UV, somehow UV helps plants tolerate desiccation. And so I looked at these gene families and several, also these are like GO terms for photomorphogenesis, response to light, stress, and photosynthesis. None of those turned out with any significantly different distribution. This above the line is above and below the line is log fold change, and the red would indicate that the whole group is significantly different. And both Ellipse and Leas were significantly different as a group with UV treatment. So this was the first time to see that these genes are not just important in desiccation, but are also involved in the UV response. And so to try and translate that, this is what I think, without UV, there's an increased stress of desiccation. And that's what we were seeing in why these plants were coming out of the field, where they still had whole years worth of desiccation events, but they didn't have UV. They came out less healthy, less prepared for photosynthesis because they were damaged by those by those desiccation events that they were exposed to. And these clues, it's all sort of lining up because empirically, if you just take some mosses of all around the world and test them for desiccation tolerance and test them for UV tolerance just by blasting them with UV or drying them out and seeing how they survive, the two tolerating UV and tolerating desiccation are correlated among mosses. So there's something going on there, but still trying to work out all the details, but Ellipse and Leas are involved here for sure. And that was basically it, but I'll wrap everything up here because I did go through a lot. Mojave Desert's quartz rocks provide a distinct microhabitat for hypolithic mosses, including very low light, very little ultraviolet radiation, buffering from temperature extremes, and longer hydro periods. So these are things we've learned about that study. Also, hypolithic Cetrichia Canada plants grow longer and greener than soil surface plants. And UV radiation induces a flavonoid sunscreen in this species. And finally, natural levels of ultraviolet radiation have beneficial effects on photosynthetic biology. Don't quite know how that works out yet, but we know the benefit is there because there's increased evidence of increased stress with removal of UV radiation. And finally, this species undergoes sustained thermal quenching, where it releases excess light as heat for days after rehydration. And ultraviolet radiation inhibits that recovery. Those were always a little bit lower than the field collected plants. So in summary, UV radiation may induce a response that protects from other stresses, such as desiccation, which is, it's kind of like you do a lot of research and find something that someone else already found, but in a totally different system with a totally different question. So it's both surprising and not, I guess, is what I'll say. There is somewhat of a mind, what is it? We're starting to change how we look at UV radiation. So it traditionally was really only looked at as a stressor, and now it's starting to be considered a eustress, a beneficial, a good stress, in that plants need some amount of stress to be able to tolerate other stresses. So, and I think my results really are consistent with that kind of mindset shift. So just to give you a little clue about what I'm doing now, I would love to follow. I haven't done this yet, actually. Next steps on that project, follow up on the identity and function of the sunscreen pigment. I have this class, like a flavonoid, but that's all. And that, even needs confirmation. Currently, I'm looking at evolutionary history of this group in using 600 samples of Cetrichia from across the world, and trying to understand what I'm trying to test here is whether Cetrichia diversified with drylands in the Northern Hemisphere. It's because they have a high number of desert specialists in the group. And I hope to test for seasonal variation in photosynthetic biology in this genus, as I mentioned. So sorry for using up all the time here. Must acknowledge my funding sources, the 3D Moss group. I received a lot this was like my whole dissertation basically, received a lot of funding over the years from the California Botanical Society and California Plant Society and many organizations. But of course, I couldn't do this without my collaborators and all of the student researchers that contributed to the projects. Named here, I can't go through them all, but yeah, I think that is everything. Thank you, and I'd be happy to take questions if there's any time.
Out of time, so if anyone needs to run, feel free. But if you'd like to stick around for a few more questions, we'll either take some on Zoom or some from the audience. Okay, go for the Zoom questions.
Question one, how can hypoliths not survive for months without light in Antarctica? I am thinking about not being able to
In Antarctica. Well, I suppose it depends if they are dry or not. If they're wet for months, then I don't know, because that would be a problem. But if they're dry and frozen counts as dry, then it's a form of desiccation tolerance, and they don't need to do photosynthesis during that time. They're not growing, they're not doing anything. So it's kind of it's a different strategy of life, really. Just don't exist in the hard times, and come back when things are good, basically.
Question two, sorry if I missed it, but how does buffering
Well, I don't know if it's the temperature. I don't know the effects specifically temperature buffering has. I think the phenotype I'm seeing is mostly a function of the humidity, but you really can't disentangle humidity from temperature, because the warmer it is, the more it'll dry out. And so I would say probably through its relationship to the hydro periods, in that if it's not so hot, then they won't dry out as quickly, which means they have more time to grow, which means they'll grow bigger.
Question three, your hypolithic moss goes to any stage of hibernation, and how do they reproduce, and can spores suppress low rocks?
Okay, so they probably do dry out. We've seen some dry out, so they do for sure. They'll still go through the summer. There's just not enough water in these habitats to last the whole summer. But they probably have a shorter, as I mentioned, when we found them, it was 2 weeks after a rainfall, and it was hot. It was like 90s at least. So things don't stay wet for 2 weeks in the spring or summer in these habitats, but they did under the quartz. So they get, quite longer time being wet. But they still will dry out eventually in the desert. We found one that had a sporophyte under the quartz, and I don't think it would be a very good dispersal. Um, but yeah. So I don't know. I mean, I think that these are kind of prime species to take advantage because they don't sexually reproduce very often anyway. They don't rely on it much. So yeah, I think they're fine to do without, and they just clone their way around.
Crazy question, but I mean, are these just glacial relics that are hanging on asexually from 12,000 years ago when it was a lot wetter? Or do you see spores regenerating? I mean,
That's a good question. We do see sporophytes, and we see, you know, the sporophytes, some of them get aborted and they never produce spores. Some, and but we do see mature capsules with spores. But attempts to germinate those spores are super variable, and it seems that a lot of the spores are not viable sometimes. And so, you know, the probability that those spores are viable and then land in a place where they can germinate, who knows? Honestly, I don't know. It seems like it's a long shot, but then again, if you do population genetics on these plants, you do see evidence of sexual reproduction and gene flow across populations. And so the tricky thing is they can live a really long time and they can clone. So a single shoot may not be that old, but that genotype can be extremely old. So we don't really know the time scale of what the sexual reproduction we're seeing evidence of in the population genetics. When did that occur? No idea. So it could be very infrequent, but frequent enough that it keeps populations diverse on their time scale. It's just something bigger than our own.
Anyone else? Okay. Well, let's thank Jenna again for a great talk. And
Thank you.
after a little break, we'll be meeting up at Public House for some drinks and some socializing. So feel free to join us if you can. Otherwise, have a great weekend. And thanks all for coming.
Thank you, everyone. This was fun.